process necessarily. There is adhesion, and in fact, I have an adhesion demonstration here. We've lost most of them already, apparently, but I had 4 pens, so we'll set them up again. They're stuck. So they're adhering, and basically they weigh about, what is that, 5.5 grams, I think. I measured it. It's actually almost exactly the same as a quarter. It's ridiculous. This is 5.6 and this is 5.5 or something. It's very, very close. So, come on. Well, there's actually double-sided tape on the bottom of this. You can't see it, but I can feel it. So, come on, stay. All right, so we've had, I've been talking for a while. And we have, I'm also going to be stomping about on the stage. And if I stomp around too much, you could probably have them come down. And that's partly because there's extra forces. There are forces on those bonds. Basically, what's holding it there? It's actually, we basically got, there's something sticky. But that's not really the answer. I mean, essentially, what does that mean? Why does it stick? Why is stuff sticky even? This is sort of a question of, it ultimately underlies also friction. You know, why is there friction? You move something across a table, it requires a force to do so. And we say in physics class, oh, it's friction. And that's very easy to say, and actually we do it a lot because the math isn't all that hard. It's easier than air drag, which is why we tend to ignore air drag in all problems. But it's mathematically easier, but what is happening at the molecular level is a lot more complicated. And you have basically you have polymer molecules that are on the tape, and they interact via usually what are called van der Waals forces, which are basically dipole-dipole interactions. It's the same sort of, it's not quite the same thing, but if you rub a balloon against your head and stick it against the wall, you can get adhesion that way because basically the balloon is charged and it polarizes the wall, which ultimately, through a complicated calculation where you need to sort of draw things. You can see that the negative charges, if it's negatively charged, the negative charges are a little farther away. The positive charges in the atoms are a little closer, so that tends to cancel out and give you a net attraction. So basically the balloon sticks to the wall if there's not, I didn't bring a balloon, which would have been good. But essentially that's what's happening here, and we haven't had any come down yet. But basically if we observe those for a while, they'll eventually give up. And so one of the interesting things about the kind of bonds that you have that bond here is that they're actually rather similar. They're basically proteins or sugars that are long and a rather complicated structure, and they bind specifically to each other. So there's pairs of these bonds. They're large biological molecules, thousands, sometimes millions of atoms together, that somehow or other reproducibly assemble into these amazing proteins that will very specifically allow, say, a white blood cell to latch onto a bacterium and engulf it, or to latch onto the surface and move. So they can stick to the surface by means of these proteins. And so in a way, it's sort of like that. There we go, that's one. Now, that was just sitting there, apparently. And why did it come down? It was its time, I guess. I mean, they don't all come down at the same time. Obviously, they come down at different times. It's kind of like a random event, essentially, that they'll give up, they'll break up, basically. And bonds are sort of like that. In fact, I have this whole sort of transition theory of relationships, because you can sort of make a pretty good model of how bonding works in terms of, pair bonding. So, you're wandering about and you encounter some other person and maybe you don't get together even to the dating stage. But you have to encounter each other in order to become boyfriend and girlfriend, and then you're boyfriend and girlfriend, and there's a rate of association for that, where you form the relationship, and then they break up, and so there's a rate for that too, and eventually there's some those couples that you'll have forming and breaking of relationships, and then eventually you sort of go into a deeper level where you're bound to you get married. And then, there's a rate of formation for that, and of course there is unfortunately a rate of dissociation for that as well. And so that is a natural process, and it turns out that you can model out all this stuff. You get to know my password. Nobody steal my iPad. This is being filmed, isn't it? Yeah. Great. Okay, the formation and breakage of relationships or bonds. There's a rate for all these processes. And so the interesting thing here is, and we'll sort of stick with the bonds that I've got here, because these aren't breaking very fast. They will break. If we reset them, they would eventually come down. So what we can do is try to up the ante a little. And for this, we're going to add quarters, which are exactly the same mass. So now instead of having just this, because basically there's a force on these things all the time, it produces a certain rate of dissociation. I'm going to add one of these. Well, let's see. I'll add a couple. We'll maybe leave our third one there. All right, so we're going to just bind them on. Double-sided tape is a wonderful thing. It's a great tool. I use it in the lab. As I also use clear nail polish, which is another great scientific tool. I knew somebody once who had assembled something using clear nail polish, and which they had applied to this tiny, tiny cantilever with an eyebrow. I was impressed by that. Okay, so we've set that up. This is pretty robust. I mean, that's still. So we'll leave this here to hang. They are not coming down immediately, but we'll sort of see how long it takes them to come down. And I am sort of creating the Brownian environment with basically my motions on the stage, because I am shaking things up a little. And if I jump up and down a bit, okay. It's actually fairly robust. It's well vibration isolated. All right, that's good. So anyway, this is bonding, and we're interested in how adding more mass accelerates, how it changes the rate of dissociation, because it was very slow before. Minutes, like how long's it been? 25 minutes, and I set those things up about the hour. So we had one come down. Now we actually had 3 or 4 come down while we were waiting for here. So I don't know why you guys being here means that they've slowed down. I don't know. Except, of course, I tend to tell my students it's the inherent perversity of inanimate objects. They will try to make you look bad. So, the experiment works great until your supervisor comes along, and then it doesn't work and doesn't work and doesn't work, and you're complaining, and then your supervisor comes along and it works. I tell them it's the inherent perversity of inanimate objects. Okay, so we actually can model this situation. It turns out that you can get this rolling. So here we have little particles that have been coated with an adhesion molecule. I guess I'm going to have to show that again. And they're interacting. They're actually rolling along pretty well. That one was just hit by something. There we go. We lost one. Considerably faster. So they are moving. We can actually reproduce this in a dead system. This is a glass slide with adhesion molecules coating it. And the same adhesion molecules that are responsible for white blood cells rolling. Thanks to the biologists, we know now what those are. They were discovered in the 80s. And we can model this process with adhesion. So we still have one. One. Yes, we lost one. I saw that. Yeah. So there's also, that's one kind of behavior you can get, and actually maybe I'm not all that interested in the other kind. But I did experiments before I came here, and then also somewhere when I've been here, trying to look at basically this force dependence of reactions. And I have one technique. This is a micro cantilever. It's basically a long, thin fiber of glass. It's actually probably doesn't look all that different from this, just much, much smaller. So it's about something like 4 micrometers in diameter. This bead here, I think, is 10. So that gives you some idea of how big this is. It's small. And it's being held in a micropipette. And micropipette aspiration, this is sort of my lab's forte. We have the optical tweezers, but we even more commonly use so let me show this. Essentially, these experiments were incredibly dull. Basically, you tried to design them so that this is coated with one adhesion molecule, and that's coated with one adhesion molecule. And you bring them, and you bring the bead into contact with the fiber, and then you pull back, and yay, we got an event. Basically, we got sticking, but most of the time we got no sticking. And so and the next time there was no sticking. So essentially, this is an example of an adhesive event. And by collecting these, we can look at the lifetimes of the bonds and basically look at the lifetime as a function of force. And we can get a relationship between how force speeds up the dissociation. So if we tracked all those, the lifetimes of those pens coming down with and without quarters, then we could get the force dependence of reverse reaction rate for these pens. And I'm not sure that would be very interesting, but it could tell us actually something about the interaction between the plastic tip of that pen and the glue. And that would be a polymer process because the tip of the pen is made of plastic, and it would be probably van der Waals interactions. Okay, but that's one of the things we did. Now, we had these flow assays, just to go back to this one. This is done, oh, there we go. That was the other one. So we're now down. We actually still have one of the other pens left. So we'll see how long that one goes. That's the one without the extra weight. And I suppose we could set them up with 2 quarters, but no. Okay, so this is done in what's called the parallel plate flow chamber. And the parallel plate flow chamber, if you'll forgive me, let's see, we're going to try to go up. There we go. All right, there we go. Come on. The parallel plate flow chamber is, thank you, 2 plates. So this is a flat plate here, a flat plate here, and basically you flow liquid through it. And basically, if you look at a side profile, basically the flow is fastest in the middle and 0 at the walls, just like it is in the tube. And you put cells in here, and basically they're driven along by a force, and they also rotate. So the force both pushes them along and rotates them. And they have bonds in here that form and break. And so we have this is geometry you can simulate that in the parallel plate flow chamber. And with that, you can figure out a lot about what's going on. And have you Dr. Getz here? Doug Getz? Well, anyway, we have experts who do this kind of thing here. All right, so let's see, go back to this. All right. So we got interested in trying to do that same sort of thing, but in a different geometry. And I'm going to actually, let's see, I wanted to show that geometry a little bit here. Yeah, that's your parallel plate flow chamber, the top there. In lung capillaries, you have a somewhat different thing. You have a very, very narrow vessels. And this is a cast. Better perhaps not to think about exactly how it was obtained, but this is a cast of the capillary network, the very smallest blood vessels that surround the air sacs in your lung. So your lung comes down, it branches, it branches, it branches, it branches, it branches, it branches many times until it reaches air sacs, which you can sort of see that there's a sort of circular opening here. There's a layer of cells that cover each of these. This is the inside. It's a cast of the inside of the vessel network, and the rest of it's being dissolved away. And you get this incredibly tortuous network in here. And it turns out that this is where a lot of your white blood cells are at any given time. Because basically they have to squeeze themselves down. And they squeeze themselves down into this network, then they circulate through here. Your red blood cells are collecting oxygen here. This is why it's arranged like that. But your white blood cells also have to pass through because they're going through. And they also can get stuck. All right, so let's see. Come on. Oh, it's still in here. Forgot. All right, where are we? So I actually have, this is not mine. It's generated by the graduate student that started these experiments. And he gave me this. He eventually did some of that intravital microscopy, basically looking at it inside an animal. This is apparently a lung, probably a rat lung. And we died. Which means you get to see my document, my directory structure. Okay, let's try that again. All right. So I'll have to show this a few times because it's not immediately obvious what we've got. But basically the red stuff in here, and actually, I don't know, we got a lot of lights on here. Can you see that? The green stuff is the cells. And basically, we'll start off at the beginning. There's a cell that's sort of around here, which sort of arrives, goes through one of those narrow segments, and then moves off. So there, it's stuck, and it flies off. And basically, we see exactly the kind of behavior that you would get in the neutrophil capillaries, in the lung capillaries. So we did experiments where we said, well, micropipettes. And you saw a micropipette in the adhesive event slide. It was holding the bead. So basically there was a bead that was 10 micrometers. And I don't know, maybe I could pass this around. This isn't yours. I didn't bring any of my own students' cells, micropipettes. They would kill me. But basically, yeah, you could pass. There's micropipettes in there, and they're basically extremely long tubes. They're long, thin, skinny tubes. And, well, I might as well show you what happens. We are looking at a white blood cell. That's kind of very quick. My student would suck them into the micropipette. So the micropipette is the tube over here. And the cell was sucked in. And so we would rapidly suck them in, and then we'd see how they moved. And we could coat the insides of these micropipettes with different things. So let's see. If we coated them with something that they didn't bind to, they just kind of they trucked along, nice and slow. But if we exposed them to something that they did bind to, then once they got going, they would very slowly move. So it's clearly moving. It's not moving very fast and way less fast than it was if there's nothing to bind to. So by changing the amount of stuff that we bound to the inside, we could change the speed. And that's basically because there's a lot of bonds that are happening. And these white blood cells can interact with the molecule that we put on the surface here. It's the same thing that we had more or less the same thing as what we had in the parallel plate flow chamber video that I showed you. Same set of molecules. And so we can control that. And it's also possible to get the cells to stick. Hang on. Let's go. Yeah, this is so this is a cell that I think. it kind of stops. It's going along, and then it sort of picks up, and we can get it to firmly arrest. And we used to sometimes find that these cells would get activated because they're white blood cells. They're a little touchy. And where are we? But they would arrest, and it wasn't because the flow stopped. For instance, we aspirated a red blood cell here. These were collected from blood. We collect blood from volunteers after an IRB protocol. And basically, red blood cells are amazingly flexible. Even though that looks like that white blood cell is blocking the way, the red blood cell can get through, and it's trucking along quite nicely. There's definitely flow there. There we go. There was our last one. So lifetime, like 40 minutes. It's less than that because on average, maybe 20 minutes. But basically, we have, and we also saw some really bizarre stuff. This unfortunately turned out to be a bit of an artifact. But anytime the cell became activated, so we weren't properly treating the inside of the micropipettes, and we were quite excited when we saw this because it was the first time we'd done neutrophils, or white blood cells. We'd actually done cancerous versions of white blood cells for a while, and we saw this kind of thing, and we're going, wow, that's exciting. But it turns out that this is what happens when the cell gets activated. So white blood cells, they circulate generally in unactivated form, but they can get activated. And this one can't make up its mind which way it's going to go. It goes in, it goes out. There is flow here. It's sort of like the red blood cell. It's going past the thing all right. It can crawl upstream, no problem. And in fact, it can crawl all the way out of the micropipette. It decides it doesn't really want to be there. And notice it also has a kind of that uropod that you had before in the picture of the macrophage. Sometimes I've got other pictures, rather a lot of pictures of that kind of thing. We've also done this with cancer cells, where we've aspirated them and looked at how they also travel. And it turns out one of the interesting areas of adhesion is that you get, if a cancer sort of stays in the same place, if it basically is part of a tumor and stays there, you can go in with surgery and get rid of it. But if the cells start to come loose and start to move around the body, then you get into bigger problems. And so that's a process called metastasis. And generally, it involves the cancer cells coming off and going through the circulation, at least for a while. And so what we're interested in is we squeeze these cells into a micropipette. That's the squeezing part. I don't have a video of this, I'm afraid. And in fact, I wanted to have this. You know, they make these things, the baby toys that sort of have rods, and they used to anyway, rods and sort of strings to connect them so that you get what's called a tensegrity structure. Buckminster Fuller studied this. And it's sort of a model of what you have inside the cell. The cells are not just bags of water, they're actually full of structure. They have a nucleus, which is also fairly undeformable, and then they got other stuff. So anyway, that's we are trying to measure also squeezing and how deformable things are. Red blood cells are incredibly deformable, but their properties have also been measured pretty accurately, and we're not breaking great new ground with that. But the cancer cells, it's new. So I did want to talk a little bit about the optical tweezers that we have. So we have basically, you can use the optical tweezers to apply force. And so like the cantilever, the big tube that I had, it's a spring, basically. The more you stretch it, the more it pulls back. And so I'm going to start this off here. This is actually, I think it's just after, I think it's the same 3 cells that I was initially looking at when I showed you Brownian motion. I recorded this morning. And then I turn on this thing here. That is a laser. Basically, there's a laser which travels around the table and goes into a microscope. And actually, I really need to show you a picture. Okay. It could be worse. technical problems. All right. All right. Come on. So this is our optical tweezer setup. It's a Nikon microscope. This is basically a microscope. It's got the eyepieces are here. The objectives are under here. It's a so-called inverted microscope. And the sample sits up here. It's basically, may not be able to see it, but basically it's a glass cover slip, 2 strips of double-sided tape, which can cut down the middle, and the straight ends have been laid side by side to make a little channel. You basically stick stuff in there. You can get water to go in by, or stuff to go in by capillary action. So you put in milk and beads, for example. And then we turn on a laser. So there's a laser here. This is the top view of the microscope. The laser travels around the table. This is not a picture. Lasers, of course, you don't see the light here unless you're actually looking down in the beam, which since it's an 800 milliwatt laser, you don't want to do. And infrared, too, you can't see anything. So we use little sensors, basically, to steer it around. There's what's called a beam expander to take the narrow beam that we get and make it a big beam. We then have various mirrors that, slightly out of view, bounce it around and off this plate here. And with that plate, we can sort of control our reflectivity for the laser beam. And the laser then comes in through the microscope, bounces off a mirror here that bounces infrared light up and lets visible light go down through. And so basically we can trap in here. And there's a lens that basically brings the light to a really tight focus. So having seen the picture of what that looks like, you can see what it does. So we'll start this again, hopefully not dying. So I turn the laser on. I don't have the laser on at the moment. There's a shutter on it. I've got it off. Ah, it's on. We actually have a stage on this microscope that can be computer controlled. So we've got a little joystick. Sorry? Okay, I think that was up at the desk. Okay, so anyway, I picked up a bead. You notice it's doing a lot less Brownian motion. That's basically because it's attached to a really big spring. And so in order to go, it can't go very far from its rest position. It's still doing Brownian motion. I lifted it off the surface. So basically I took the focus, I went focus up, and I think that's all you got on this one. Sorry? Sound back there, distracting. Okay, so this is a trap again. I'm going to load it up with a bead. So I've loaded it with a bead. I lift it off the surface again, and other stuff comes in. Unfortunately, they trap. I wouldn't normally have all that much stuff in there. And then we're going to take it for a ride here. I think this is the one, yeah, different speeds. So that's 25 micrometers per second. Nothing. Actually, it would be slightly displaced from its rest position, and we can use this to calibrate the trap here. Basically, it's sort of like this. The more you bend it, the harder it pushes back. It's Hooke's law, which is one of the most amazingly useful things and still so true. So that was a little faster. We got another fast, I think it's 75. We got another bead too. Actually, that's good because we blow some of that out in going there, and but we got a few more things. Yeah, and that's it. So I did manage to blow some of them out with the hydrodynamic force. There's actually a force from the fluid drag, and basically this can be easily calculated. The math is not that hard, and fluid drag is pretty easy to include. So we now have basically that trap, and we know that there's a bit of stuff there, so I turned off the laser. I know there's a trap there, because now when I let it go, it's doing Brownian motion. And the various constituent stuff goes its separate way. All right. So I got another trap here. It turns out we can also have multiple traps. So I can create this trap here, the green circle, and I can move the bead around. I can also grab this one. The trap is not as strong, and the more traps I get, the weaker they all get, except for the central one, which is always there. And I pulled that one. I got it a little too close to the black hole, and it got sucked in. But basically, we can pick things up and manipulate them and move them around. And the idea with this is there's actually quite a lot you can do with this. And it's been used to, and I'm pretty much at the end of what I've got. But you can use this to do some things. Because basically what this is, the optical tweezers, is it's handles. I mean, the bead and the micropipette, their bead and the microcantilever, they were also handles. You have the micropipette to hold on to the bead and move it in and out. And the laser trap is handles. And so you can take a stretch of DNA, say, and it's not so easy to do this, but there are some, DNA has very interesting mechanical properties. It has a tendency to get twisted. When DNA gets red, that tends to twist it a bit. And when it, in the normal course of daily life, DNA is sort of wound around all sorts of things. It can sort of twist up like that. And in fact, if you still have an old phone cord, you might have seen it do this. Basically, it relieves the strain by doing twist. And it can also, it works better if I've got a paper, but basically it can also make little loops. And in fact, you can get those wrapped around things. And in DNA, it tends to get wrapped around what are called histones and make, DNA packages itself up really well. But it's like there's a meter of it in every cell. So there's something like that much in every human cell. And it's packaged into a nucleus that's smaller than these cells here. And that's possible because the DNA is incredibly thin. It's only about a nanometer in diameter. So the volume is such that it can fit in, but it needs to be really well packed. So we can do this. We can actually stretch DNA. We can take the DNA, which has a natural twist to it. We can stretch it by basically taking one bead in one trap and another bead in another trap and stretching. Of course, that can happen. But usually what happens is the DNA is kind of twisted up, and you can sort of untwist some of the more complicated strands, since most DNAs, if you look at them, are sort of like this. So you can definitely look at the... Okay. I would say that these things here are bonds to where they get weaker if I apply more force. There are bonds where that get stronger if I apply more force. And the classic example is the finger prison. I can get my fingers out of this reasonably easily if I don't pull on it. But if I pull on it, basically there's a conformational change in the prison that means that it's a lot harder for me to get this out. And basically, the harder I pull, the harder it's going to get. And it turns out that the bonds that hold these cells together are kind of like that. So if you use techniques like the microcantilever or atomic force microscope or the optical tweezers, you can pull on these things. So yeah, finger prison. This is what's called a catch bond. So these things are slip bonds. The more force you put on, the faster they break up. But it's also possible to conceive of something like, for example, here, if I couldn't find something that was really like a finger, I've got a 10 cc syringe plunger, which is pretty close, but it doesn't grip as well as a finger does. And so I can't sort of get it to do that. But I could probably get it, the more weight I put on, the longer it would last. And okay, it's about 1:45. And I could talk more, but maybe it would be good if there were any questions. Yeah. Yeah. Well, so with the micropipette, do we control it manually? Well, actually, I have done, in making microcantilevers, the way you do it is that you look through a microscope and you look down at a surface that's got a lot of little cantilevers, and you take a micropipette and you pick one of them up by static electricity and then lift it up, and then you stick it into the mouth of another micropipette. And there are other ways to do it, but this turns out to be the easiest way. It's not easy, but it's easier than, and you can make these at a rate of about one every half hour, but you're basically manipulating something into a tiny, tiny hole under a microscope. And actually, your finger's perfectly capable of doing this. So you could do it manually. We don't do it manually. We do it with micromanipulators where, yeah, you're moving it manually, but you can move it incredibly stable, and it moves with very small motions. And in fact, for the ones that we had there, we didn't even touch anything. It was all under computer control. And so for the adhesive event, that was all under computer control. It was done by what's called a piezoelectric stack. And basically, this is something with an electric field will change the size. So you can make things expand or contract based on what electric field you put on them. So we did that, then we would have the computer say, okay, go like this. So we don't have to do it. With the laser trap, we're moving it manually. You can tell the computer to move the trap from here to here, and it will do that. And we'll try to do it at a speed that won't lose the particle. But the traps are not infinitely strong. In fact, one of the things about the laser trap is that you need something where the forces are pretty weak, because the traps are not especially strong. The cantilever, I could get forces up to a few hundred piconewtons. But the laser tweezers, I tend to lose the bead after maybe 10.
In your experiment with the white blood cell and the bacteria, what's the prime mover there? Or in other words, how far does that bacteria have to be from the white blood cell before the white blood cell senses its presence?
That's a good question. They can sense it from a pretty long way away. I mean, the bacteria vary in terms of what they give off. For some certain kinds, I can't remember gram positive or gram negative, it's lipopolysaccharide, and that kind of diffuses off. Hard for the bacteria to hide from that.
So there's always an effusion from the bacterium that eventually the white blood cells will become aware of.
And white blood cells this is an experiment I've done, but the people who looked at migration have shown that they can climb a gradient, or they can go down a gradient, or they can go horizontally across a gradient, depending on what they have to do. So if the thing is increasing as you get towards the target, it will go that way. So. Why do you want to untwist the DNA? Yeah, I didn't repeat the other question. Why do we untwist the DNA? Basically to understand DNA's behavior, the DNA mechanics. So why might you want to do that? Mitosis involves pulling cells apart, pulling the nuclei apart, not the nuclei, the DNA. Basically, you have this whole process where strings are attached to the chromosomes, and then they're kind of pulled on. And yeah, I've got a colleague he's a chromosome puller. He's measured the stretch of the chromosomes. And so understanding how they behave can have an impact on our understanding of how that process happens. That sounds a little, if I could tell you a disease, I would, so I'm not.
What's the mechanism of the optical capillary? What's the force that keeps that within the
So the question is, what is it that does the optical trapping? So let me see if I can sketch this, because it's kind of hard to do with hand waving. So let's see if I can. So let's get a new page. So it's more complicated than this because the particles are very small and geometrical optics doesn't really work. But what we usually say is you've got a particle here, and you've got a beam of light. And the beam is very intense somewhere, and gradually the intensity of the beam falls off. So the beam is really intense here. Come on. The beam is really intense here. It comes in, bends towards the, it bends, and then it bends away from the normal here. And you get a change of momentum for the light. Light, it turns out, has momentum. So even though it has no mass, it has momentum. And so if you go from momentum in that direction, momentum is a vector quantity, and it's different afterwards. Turns out that in order to make that happen, you need a force. And so therefore, there's a force on the particle that moves it that way. And if you've got particles coming in here, they bend the other way. So you get this change of momentum. But the beam is less intense out here. So even there's a force from that, but it doesn't pull as strongly in the other direction. And the result is that the particle tends to snap to the most intense part of the beam. It also tends to snap to the focus. Although there's also radiation pressure. Light can push. So out in space, if you've got solar sails, if you could make solar sail, if you could expand a thin enough sheet of mylar and unfold it in space and make it big enough, which is a challenge, and get the mass small enough, then you could conceivably go somewhere. But there's some pushing here. So the particle tends to push a little bit off the focus. But we do have a tractor beam. We have a tractor beam for particles, not for spaceships. How are experiments used in cancer research? So we're with any cancer project, and we have a grant for this. You can sort of try to make a case for treatment, or you can make a case for diagnosis. There are certain characteristics that we expect to see in cells that are more metastatic. And so the ones that tend to go around the body and be more of a problem. And the goal is to try to understand the mechanical properties of the cancer cells that go with being more metastatic. So we can use the micropipette setup. We also could use the optical tweezers to pull on this. I've had a student try to get this working, where you could attach handles to the sides of the cells and stretch it and see how much it stretches. And the question we're trying to see is whether that's useful as diagnostic method. So yeah, the question is how is the optical tweezer used in cancer? Beth.
We have, rivers flowing and sediment flowing. We have equations for movement. How do you know if they're similar to the movement of blood cells in the veins, or there's the sticky factor which we don't have along the edges of the
So the question was flow in rivers, like in geology, where you basically have, or hydrology, where you have water flow, say, in the Hawking. How is that different from the flow in blood vessels? The main difference is that kind of technical, but there's something called Reynolds number, which governs what kind of flow you have. And actually, the no-slip boundary condition, I mean, you look at the Hawking River, it's fastest down the middle, it's basically zero at the walls. That's still there. That actually carries over. But actually, the fluid flow for when you take into account the river, the Reynolds number is large, and basically you can get turbulence. In the body, maybe at the aorta, you could get turbulence, or in the heart. But everywhere else, the Reynolds number is such that it should be laminar. Now, the only difficulty is blood is not what's called a Newtonian fluid, whereas water is. Although if it's got a lot of algae in it, maybe not. Questions back? Yes.
Is there the lateral healing becomes stronger as it is more force on it, or is it like heat on it?
So the question is, I mentioned the slip bonds where it's easier to, as they apply force, it gets easier. Then there's the catch bonds, where as you apply force, it gets harder. And the question was, is there a case where the catch bond situation keeps going up and up and up? Well, so, okay, I got a problem here. So eventually, I'm not going to do it, but if I pull hard enough, I will break something. And in fact, the interesting thing seems to be that a lot of the bonds that were involved in the processes that I showed you with white blood cells, they seem to sort of start off when their natural configuration, they're not as strongly bound as they could be. You displace them a little, they actually get slightly stronger. They last a bit longer. But then usually it collapses. You pull them a little further apart, they weaken again. So actually you're very right, nothing goes up forever. Even the catch bonds there, eventually they're going to break.
So basically the surface molecules of the blood cells will modify, right?
So
So the surface molecules of the blood cells
yes.
should modify in order to have these kind of different, whether it's a slip or catch times, right? To behave differently.
Yeah. So the question was the surface molecules for the, say, white blood cells, there can be some that maybe are slip and some that are catch.
Yeah, so depending on the situation, it has some kind of freedom to modify.
Yes, it has some, so the bonds can sort of slip go from a catch to a slip situation. It's the same bond. So sometimes it can, under some regimes of force, it can be a catch bond and some a slip bond. And that's pretty commonly found, like the previous question. And different bonds are different. So some of them are always slip. But actually, more and more of them seem to be turning out to be catch, at least over some range. The bonds that allow molecular motors to run along trackways in your cell, they seem to be tuned, interestingly. They have their peak binding at about the maximum force that they can push things along at. So about 5 piconewtons or so.
Has this been applied to hardening of the arteries? I mean, how does does anyone know how that happens?
Well, I mentioned the bifurcations where basically you go down here and it's sort of at the stagnation point. There's actually quite a lot of work that's been done on that, trying to look at the flow patterns and then also what the tissue changes are in the cells that line the blood vessels that come from the fact that you've got these complex flows. It turns out that flow can tend to align, so the forces in the flow can tend to make cells line up with the flow. Or in regions that there's less flow, they'll be more cuboidal. So in other words, they will have a different architecture. And this has been known for quite a long time, but also partly from studies like this. So you sort of take a cell, you grow them in a chamber without flow, and then you put them in flow overnight, and they go. So yeah.
Well, with that, I'll let you guys in a minute. With that, let's appreciate it. We'll open it up. We'll come up and talk to you in a minute, okay? For those of you that still have questions, you can come up afterwards and talk about your case.
Sure.
Couple of things to note.